First of all, I would like to really thank the organisers for this opportunity to speak, and I would like to take my beautiful hat off, especially to the hub, because this is an Instruct-ERIC meeting, and it's really been a pleasure to be here. All right, so I'm going to talk to you about just a little bit of the work that we do on virus entry and assembly. And the main reasons that we're interested in this are for the development of effective vaccines and antivirals. And so today this is just a snapshot on one part of that work. So I'm going to talk to you about enteroviruses. These are small RNA viruses which cause all sorts of lovely diseases like coughing and spitting and meningitis, sepsis, paralysis. And also we have a consortium working on those coronaviruses which are now known to cause type 1 diabetes. So there are a lot of infections annually, and currently we don't have treatments and we have very, very few vaccines for these viruses. So they have a high social economic cost. They're very, very simple. They have, let's see if I can work the double system. They have an icosahedral capsid, which is made up of 4 proteins, 3 that you can see on the outside, BP1 around the pentamers, BP2 and 3 are alternating around the 3-fold axis of symmetry. And then on the inside, the red protein is BP4, which is a small protein, which is very much involved in the release of the RNA. So in terms of the life cycle, this is a schematic of the whole life cycle, and today we're just going to focus really on the inhibition of this attachment and entry process, which in this case is via endocytosis, and the viral RNA is uncoated within the endosome. In more detail, we found that there are many factors actually outside the cells that can already prime these capsids for the release of the genome. One of the surprising results we had was that even the presence of serum albumin for these viruses that are circulating in the blood can actually start to trigger this activation. And what happens is that there is a lipid factor found in 60 pockets within this capsid, and when you start to release that lipid factor, you cause expansion of capsid, and that can be seen, for instance, here in a movement, localized movement at the 2-fold axis of symmetry, but actually what's happening is that there's a twisting and expansion all the way around. And it occurs in different steps. These can be receptor triggered, but in cases of Coxsackie virus A9, it's not receptor triggered. It's actually triggered by changes in the ion concentration in the endosome as the endosome matures. And one sees subtle differences in the positionings of the BP1 N-termini, which are going to come out of the capsid as part of this activation, and in the size of these pores. This means that in all of these preparations, we have a lovely heterogeneous mixture of capsids which all need to be sorted out, and when you're talking about 4% expansion in a capsid, which is around 30 nanometers, that's a bit of a pain. And also when one starts to look at specific points on the capsid, the symmetry actually ends up as being a disadvantage rather than an advantage in some cases. So we've worked on various antiviral strategies which are targeting genome release. One of the ones we worked on with the NUDES lab were inhibitors of Coxsackie viruses. We found some broad-range inhibitors, and they were targeting an interprotemer pocket. So that's basically, if this is the basic asymmetric unit of the virus, then the drug is binding between these asymmetric units. These are really quite nice, quite effective, and they are stabilizing. In fact, actually, Dave Stewart's lab has now started to use these as a way to stabilize poliovirus, virus-like particles as part of the poliovirus vaccine development. Now, our collaborators were looking in general for capsid inhibitors, and they found some, and they didn't know actually where they were binding. So another popular place to find inhibitors is in this hydrophobic pocket in BP1. So this is again the whole capsid, and then this is the protein around the 5-fold. And this is a different pocket to the interprotein pocket. So they have found these compounds from a big library that they had, unknown binding site, but low micromolar affinity, so quite nice EC50s, and they're not particularly toxic to cells. And so, in collaboration with the Pocci Lab of Medicinal Chemistry in Rome, and they went to the library and they then synthesized around 29 derivatives from this initial hits, and then Marco Mariomaki in Uvascular then went ahead and did virus inhibition assays in cell culture to find which of these were effective. And then we also checked with an RNA sensitivity fluorescence assay to see whether or not the RNA could be released or not. And basically the idea with this medicinal chemistry approach was just to see which areas on the molecule were useful, and in fact it turns out this R group was the one to target. We then went ahead with 8 of these inhibitors to see where they were binding on the capsid, and to see if we could make any sense out of the slight differences in the affinity of the compounds. And then again, the Pocci group then went ahead with molecular dynamics, now knowing actually where the compound bound, which helps. So I'm just going to show you a few of these different compounds. Obviously, from here I'm zooming in on this particular pocket, which is indeed this hydrophobic pocket in BP1. So it's a known binding site for inhibitors. And what we see is with these 3 compounds, we see that indeed we have clear density for them binding to the pocket. And what happens is that there's a very obvious switch in the conformation of this tyrosine 210 at the base of the pocket, which you don't see with the lipid factor that normally sits in this pocket. There the tyrosine is in a different position. And so these are forcing the opening of that pocket, allowing the molecule to enter. So these were relatively straightforward. They're basically between 2.3 and 2.8 angstroms resolution structures, which is also in this area. It's also similar resolution. These are all data collected in the Instruct Center in Finland. But the story then varies when you look at some of the other pockets. So in 2018, these are binding still in the same pocket, but they have a low occupancy. And we can see that by the fact that we can see the 2 different conformations of this tyrosine, and you can see, again, you have this extended density up here, which is most likely due to some of the pockets being occupied by palmitate. And in terms of sorting this out, you can go in, you can do localized reconstruction, but we're talking about identifying a small molecule which is not covalently bound to the capsid. There's 60 of these pockets in each capsid, and so the sorting of this is still tricky. But this indication of the 2 different conformations encourages us to believe that we do have partial occupancy of these pockets. Then with 16 and 14, there we see that we again have this partial occupancy, and now we have even lower occupancy than with the first compounds. And so we're seeing gradation of the occupancy. So then, if we just compare 2 of these compounds, we have the EC50 down here, so 0.97 and 2.13. These were then the project group then did some molecular dynamics on these, and you can see the comparison between in yellow, the cryo-EM positioning of the molecule, the MD simulation in pink, and for the compound which has the best binding, what you see is a base stacking of these tyrosines here, so tyrosine 146, which allows the molecule to sit further and deeper into the pocket, and where we have less efficient binding, then the molecules are moved downwards, so they don't have that same affinity. And so it was a nice, clear result after looking at a lot of structures. And so in conclusion, what we actually managed to do was to work out what was the targeted area on the capsid for these inhibitors. We know that it stabilizes the capsid and prevents genome release, that's the mechanism. Of the 29 synthesized derivatives, 10 had EC50 values between 0.68 and 14. And of these, 7 had CC50 values, which were higher than 200 micromole, and so those are the ones that we then studied. The base stacking with this tyrosine 146 in the hydrophobic pocket is a key to increase potency. And what we can also tell from this is that you don't need to occupy all of these pockets to prevent the expansion of the capsid, which is good. So even if one has really poor solubility, you can still inhibit these capsids. Finally, I would like to thank lots of people. So this has been the part of the thesis work of Zlatka Blavesh, who has done the majority of the structures in here. She was helped by 3 master's students, Chris, Valtteri and Sienna, and Ausra, as a senior researcher in the lab, was overseeing a lot of this work. Benham and Pasi were collecting data. University of Uvascular, the work on the virus inhibition assays was done by Varko, Laura and Mira, and big team in the University of Roma, in Rome, who were really the medicinal chemists who went ahead and made all these compounds, did a huge amount of NMR, which I didn't show, and all the molecular dynamics. And then I'd also like to thank Marta Caroni. So there was one structure here which was collected in Sinai Lab. And I'd like to thank you for your attention.